Welcome, everyone. Hi, everyone. Welcome. We'll just give folks a minute to get connected. Hello, welcome, everybody. All right, great. Welcome and thank you very much for joining today for the scientific update on HPAI, our flock talk. Just before we get started, I just wanted to cover a couple of housekeeping items. This webinar will be recorded, so for folks who would like to go back and listen to sections or to share the information with your colleagues, you will find the recording of this webinar up later today on the web page for the Canadian Association for HIV Research, CAR, and we will send the link along to all those who have registered to be able to find the recording. The speakers are going to join us here in a moment to get started, but just wanted to let you know that if you have questions along the way, please post them into the Q&A chat box at the bottom of your screen. The questions will all be answered at the end of the webinar, so please stick around till the very end for any questions. And with that, I will turn it over to Dr. Sam Mubareka.
Thank you so much, Erin. That was Erin Love from CAR. We're very grateful for a partnership with CAR to be able to present this webinar. So good morning or good afternoon, wherever you may be. Thank you so much for joining us. It's a real pleasure to welcome you to this scientific update on highly pathogenic avian influenza virus. So before we get started, we have two excellent speakers. I want to acknowledge the land that I'm joining you from here at Sunnybrook Hospital. This is where I work, and also I live in Toronto. It's treaty territory of the Mississaugas of the Credit First Nation, and also the ancestral and traditional lands of the Anishinaabe, Haudenosaunee, Ojibwe, Chippewa, and Wendat nations. Now, I appreciate that people are joining us from across the country, so it's important to take a moment to acknowledge the lands on which we all live, work, and play. Home to me will always be northwestern New Brunswick, and those are the traditional lands of the Madawaska Maliseet First Nation, and where the Living River, the Willistook, flows. So today we're going to have a more formal presentation of what has culminated from a number of very informal talks. We call them flock talks. It's a group of scientists and practitioners who came together to collaborate, share data, share ideas around how to meet this really important challenge around highly pathogenic avian influenza virus. And this group came together after perhaps the pandemic galvanized a smaller group called the Wildlife Emerging Pathogen Initiative. So the webinar that CAR is very generously hosting today is courtesy of a CIHR Planning and Dissemination grant, and also a component of my Applied Public Health Chair from CIHR and the Public Health Agency of Canada as a component of the Wild Epi Knowledge Mobilization aim. So today we'll do an overview, and we'll talk about two major changes, or I'll call them emerging issues, around highly pathogenic avian influenza virus from the last year or so. And we have two excellent speakers, and I have partner in crime, co-moderator, Dr. Jimmy Dikiakos, who will go ahead and introduce our first speaker. Over to you, Jimmy.
Thank you, Dr. Mubareka, and good afternoon and good morning, everyone. Thank you for joining us today for this very special webinar. And thanks also, let me also thank Carr and Erin Love and Andrew Matejcik for their generous hosting of this event. It is my distinct pleasure to introduce the first speaker, Dr. Anthony Signore, who is a research scientist at the National Center for Foreign and Animal Disease. His research incorporates bioinformatics, evolutionary analysis, molecular biology, protein biochemistry, and comparative physiology to study the functional evolution of viral and vertebrate genomes. Current work in his team aims to identify genomic regions associated with increased viral replication, virulence, transmission, pathogenicity, and/or mammalian adaptation in avian influenza viruses. Without further ado, the title of the talk is Surveillance and Phylogenetics of a Clade of H5N1 Viruses, the Emergence of Genotype D1.1.1, and Resistance to Various Antivirals. Tony, please take it away, and thank you for joining us and for accepting the invitation.
All right. Well, thank you very much for that introduction, and thank you for extending that information. Very happy to be here. Thanks to all of you watching. It's so nice to present in a seminar with such a well-attended audience. So as Jimmy mentioned, today I'm going to be talking to you about a little bit of an overview of the H5N1 panzootic across Canada and the broader North America. And specifically, I want to get into some details on the emergence of a new genotype that has been shown to confer some resistance to a commonly used drug to combat avian influenza infections. And so before we get too deep into the weeds, for those of you that may be unfamiliar with avian influenza viruses and are just here as a curiosity, I want to give you a little bit of background so you know what you need to know moving forward. So influenza A viruses are an envelope virus with 8 segments of single-stranded RNA as their genome. Dotted around the protein's lipid bilayer are primarily 2 surface proteins we call hemagglutinin, or HA, and neuraminidase, or NA. These proteins are primarily involved in cell entry and release from the cell, and they are also how we subtype these viruses. So there are 16 HA subtypes that circulate in birds and 9 NA subtypes. And so the complement of these 2 proteins on the cell surface gives us the subtype names like H3N2, H5N1, H1N1, etc. So influenza A viruses primarily circulate in anseriforms, those are geese, ducks, and swans, and hydroforms, so shorebirds. While these are the primary reservoirs, they can infect pretty much most vertebrate species, including humans. So, for example, H3N2 is a common seasonal influenza virus for us. These viruses, they circulate in wildlife, and as they do that, they commonly spill over into agriculture. In the case of Canada and North America, in this particular virus, it's primarily poultry farms. And with these dense infections in poultry farms, they can, and sometimes do, spill over into humans. The particular panzootic that we're dealing with has spread quite broadly across the entire planet. It's infected every single continent with the exception of Oceania. Since 2020, it's impacted almost 500 million poultry, and it has been confirmed infected in more than 600,000 wildlife cases, although given the difficulty in detecting these cases in wild species, I suspect that number is grossly underestimated. This virus first emerged in North America on the back of a black-backed gull that was detected in St. John's, Newfoundland in November 2021. And we know this thanks to an enormous partnership of Canada's Interagency Surveillance Program for avian influenza viruses. So before I move forward, I just want to take a moment to acknowledge all of these agencies that have come together and done and contributed so much to this great work. They have been a very, very busy group of people. Up to this point, almost 55,000 live, dead, and sick animals have been tested as part of this effort, and almost 10% of them come back positive for highly pathogenic H5 influenza viruses. Most of the detections have been in waterfowl and hydroforms, as you would expect, given what I just told you and where they circulate, but they also detected in a wide array of other birds, and those include terrestrial mammals and marine mammals as well. As I said, as these viruses circulate throughout wildlife, they spill over into agriculture. In Canada, this virus has impacted almost 600 different farms in Canada alone. These are entirely poultry farms. And up until this point, almost 15 million poultry have been impacted by this virus. Outside of poultry farms, there hasn't been any impact, at least in Canada, although that's not the case more broadly across North America. Early in 2024, there was a disease circulating in dairy cattle in Texas that reduced their milk production. It was initially called Texas agalactase syndrome because they didn't know what it was, so they just called it Texas bad milk syndrome. However, it came to light eventually that this was indeed highly pathogenic avian influenza infecting these dairy cattle. Up to this point, over 1,000 herds of cattle have been affected across 17 US states. From these infections, 41 human cases have resulted from exposure to infected dairy cattle, as well as a number of other human cases from exposure to poultry and other sources. So if we move back to the dogma of how the virus persists through wildlife, poultry, and potentially into a zoonotic spillover into humans, I think it's fairly safe to say that we have a toe over this line here, where we are just dipping in to human infections. But in order for these infections to persist and spread throughout humans, it requires an opportunity for the virus to evolve and become more well adapted to circulating and spreading within and between humans, which is not something we've seen yet. However, this virus, it evolves quite quickly, as do most RNA viruses, by two primary mechanisms. One is the sort of traditional, normal, an error-prone RNA polymerase creates a number of single nucleotide substitutions over time. Second method and way it evolves is something called reassortment. You have two or more influenza viruses infecting the same cell. As their genomes replicate, their genome segments get jumbled up together and they get repackaged into new genome convolutions. So it's a form of recombination, providing a whole bunch of new and exciting genetic diversity across Canada and the US and the broader world. Reassortment has been the underlying cause of 3 out of 4 influenza human pandemics. And given the situation that is very prevalent, this virus is very prevalent in poultry and mammalian agriculture farms, a situation like this that we saw in 2009, where we have a reassortment between a swine influenza virus, an avian virus, and a human virus coming together to create a pandemic. It's not a cause for alarm yet, but it's definitely something that should be on our radar because it is certainly possible in the near future, especially given how prolific this virus is at reassorting with other low pathogenic avian influenza viruses that circulate in the wild. So there are a number of low pathogenic viruses that naturally circulate in anthroforms and cadgiforms and other wild birds. This particular highly pathogenic H5N1 virus has reassorted with them to create more than 100 different viral genotypes in Canada and in the US, which provides a very complex evolutionary history, as demonstrated by this spiderweb-looking figure here. What you're seeing in this diagram is that each single dot represents a unique H5N1 genotype, wherein the arrow represents a reassortment event between an H5N1 highly pathogenic virus and a low pathogenic virus to create this new genotype. One thing that we hadn't seen until very recently is a reassortment event that occurs in the surface proteins of the virus. So just as a reminder, those are primarily neuraminidase and hemagglutinin. These surface proteins, as I said, they're not only involved in cell entry and cell release, they are the target of prophylactic drugs, they are the target of vaccines, they are the immune system's target. So up until this point, we hadn't seen any reassortment that would result in an antigenic shift until October 2024. Where we saw a wholly Eurasian H5N1 virus reassort with a North American-derived low pathogenic virus to create a new genotype called D1.1. This D1.1 virus took 4 segments from a North American lineage virus, PP2, PA, and NP, and importantly, the neuraminidase segment, one of the surface proteins involved in exit from the cell. And this virus completely took over the landscape, spread quickly and spread often. So if we look early in 2024, we see there a mix of different genotypes circulating in Canada and the US. There's a typical summer lull while the birds are nesting. And then in October 24, when this virus emerged, it exploded. It completely replaced, for the most part, all other circulating genotypes and was detected in huge numbers. Between October and December 2024, it had been detected in 24 states and provinces. First of which, that I'm aware of was on October 6th in Alaska, followed very nearly after. The very next day, it was detected by BC CDC on October 7th. And within 2 weeks, it had already made it as far south as California. And so using some phylogenetics based on the sequences collected from these viruses, we could infer how and when the virus spread. The unfortunate thing, however, is this virus seemed to spread so quickly, there aren't a lot of informative nucleotide substitutions to make those inferences, so we can't actually say with confidence how the virus spread across every single state and province where it was detected. There are a few things we can, however, say with confidence. The virus appears to have emerged in BC despite the earlier detection in Alaska. There is still some uncertainty there, but we see well-supported transitions from BC to Alberta, to California, and to Kansas. And from Kansas, we see a number of well-supported transitions that spread the virus across the southeastern United States. We can provide a similar phylogenetic tree, this time that helps us infer not jumps between states and provinces, but jumps between species. So from this particular tree, it's colored according to the origin of the viral species, be it domestic bird, human, wild bird, or mammal. We see that domestic birds and wild birds, as you would expect, dominate these detections, and we can infer multiple introductions from wildlife to poultry. We see multiple instances of farm-to-farm transmission as well, so slip-ups in biosecurity. And then in Canada, at least, we just from October until now, we had 94 different poultry premises across 9 provinces infected with just this particular virus. And then we see the one and only human case that has been detected, fortunately, in Canada. It was a fairly severe case. The patient eventually did recover and was allowed to leave the hospital, but it looks as though, at least based on these data, this case was wild bird in origin. However, there weren't any concrete epidemiological links found between the case and other investigations surrounding that person's home and other epidemiological contacts. So one thing we see in this particular tree, in addition to inferring transitions between species, we saw the emergence of a fairly rare amino acid substitution. So virus was collected on a poultry farm near Abbotsford, BC on November 1st, 2024, revealed an exceptionally rare amino acid substitution. From publicly available isolates on GISAID, only 8 out of almost 18,000 of them carried this particular substitution. It's in the neuraminidase protein, so that protein that is derived from a North American lineage virus due to the reassortment event that created genotype D1.1. And this confers strong resistance to the neuraminidase inhibitor oseltamivir, marketed as Tamiflu. It rapidly spread across 12 poultry farms in British Columbia, but unfortunately has not been detected since November 30th, 2024. And more fortunately than that, is it not detected in any wildlife species. So the spread of this virus was simply farm-to-farm transition. There were no wildlife intermediates. And so from these viruses, and with the help of our collaborating friends across the hall at Canada's National Microbiology Laboratory, we performed some tests to see if this particular virus carries the same phenotype that has been published in the literature. This is a completely different virus. Despite this mutation being fairly well studied, this is a different virus with different background relative to what's been published in the literature. In the literature, it dictates that the wild type represented by this molecular model in purple, oseltamivir comes in to the active site of neuraminidase and blocks its activity. The substitution at 275Y, dictated by this gold-yellow color, the larger substitution of tyrosine in place of histidine pushes that oseltamivir out of the binding site, theoretically reducing its efficacy. And so we ran tests on these samples collected in BC poultry in 2024, and they did indeed find that the occurrence of this substitution at 275 with the tyrosine in place of histidine dramatically increases, almost 200-fold, the IC50 of oseltamivir effectiveness. Fortunately, another drug, zanamivir, is not affected by the substitution, so there are still means to combat infections should this mutation spread to species outside of poultry farms. Well, what's interesting is that this substitution emerged in the first place. Typically, we don't see this substitution, although it is common in human infections, we don't see it in the absence of oseltamivir administration to the patient, because this particular mutation actually makes the virus less fit. It reduces the overall stability of the neuraminidase protein, resulting in fewer of them being created. So it makes the virus less fit in the absence of Tamiflu. So it's curious that it actually emerged in the first place. However, if we look at the neuraminidase protein that this D1.1 genotype carries, we see that it has 4 permissive mutations, 3 of which are actually found in other H5N1 proteins that are circulating in North America, but one is unique to D1.1. So it's possible that this genotype D1.1 may be pre-adapted to carry this particular mutation, as it has permissive mutations that negate the fitness effects of carrying it in the first place. However, these particular mutations were identified in a completely different protein. They were identified in human H1N1 viruses. So given that this is a very different genetic background, we don't know if these permissive mutations carry the same phenotypic effect. So our work now is starting to focus on identifying permissive mutations, if they exist, in particular in these D1.1 viruses. And we start that by looking for mutations that alter protein stability, because the H275Y substitution reduces protein stability. That's why it reduces the overall fitness of the virus, and mutations that increase stability can negate this. So we do this by starting with a phylogenetic tree. Initially, one that represents essentially the entire history of N1 neuraminidase proteins over time. And we can see from this tree that clade 2344B viruses are polyphyletic. This is because, as we know, there was a reassortment event that swapped the neuraminidase protein in D1.1 viruses, making it, providing it with a North American origin neuraminidase rather than Eurasian origin, like we see in all other clade 2344B H5N1 viruses. And so from this tree, we can reconstruct the amino acid states across every single node in the tree. And from those, we can build protein models, not just at the ancestral nodes of the 2344B viruses, but at every single ancestral node in the tree, and use those protein models to estimate the relative stability of every single protein at every single tip and ancestral node in this particular tree. And from those data, we can see that the ancestor of the Eurasian-derived 2344B viruses is inherently less stable than that of the neuraminidase protein found in the D1.1 proteins. A more negative delta G, or change in Gibbs free energy, confers a more stable protein. And from these data, we can use genome-wide association to see which amino acid positions are implicated in altering the relative stability of these proteins. And from those, we can also see the relative contribution of each amino acid substitution towards the Gibbs free energy. So we can see that a substitution up here, highlighted in red, it has the greatest statistical significance as being implicated in altering the stability of the protein. It also appears to confer a almost 2 kilojoule per mole drop in the delta G, conferring a 2 kilojoule stronger stability. And as it turns out, this particular substitution is indeed found in D1.1 viruses and not found in other H5N1 clade 2344B viruses. This is, of course, it's all done in silico, and it's still essentially theoretical at this point, but now we're moving to the process of taking all these substitutions, taking the ones that these analyses are highlighting as interesting, putting them into cells and testing them in vitro in the laboratory. And then eventually that will add to our ever-growing database of amino acid substitutions that we need to watch out for in an effort to keep the jump from poultry to humans from happening. And so with that, I would like to thank all of you for your attention. And once again, an enormous number of people and agencies that make this work possible, from surveillance, collecting the samples, all the way up to sequencing and further analyses in the laboratory. So thank you to all those people and for all your attention and for the opportunity to speak here. And I will be happy to take any questions you have in the chat or at the end. Thank you very much.
Thank you so much, Tony. That was so well articulated, both the overview and also the new data that you presented. And I would just take the opportunity here. I have to admit, when I first heard about this, I thought, here it is. This is the pandemic. This is the very beginning. And I just want to draw attention to the fact that we do owe a big debt of gratitude to the individuals who stamped out that virus in those farms in BC. That containment was absolutely critical, and I don't think we recognize that often enough. So I just want to just underscore that as a significant pandemic prevention measure, that it really could have started on our doorstep with that oseltamivir-resistant virus. So thank you so much to everyone who's on the ground there. And next, I'd like to introduce Dr. Yan Zhou. She is a molecular virologist and a very well-established influenza researcher at the Vaccine and Infectious Diseases Organization, or VIDO. Her work is focused a lot on influenza virus pathogenesis and host adaptation. Her team has been looking at viral determinants of disease severity as well as interspecies transmission. She's also done a lot of work on host evasion, and I know she's turned her hand also to vaccine development. And really, the work that she'll be sharing today is incredibly unique, at least in our national landscape, because the ability to do this type of work in large mammals with such a hot virus. So, as many of you know, influenza virus is level 2 if it's human, but for highly pathogenic avian influenza virus, it's a security sensitive biological agent. So that introduces a whole number of complexities that Dr. Zhao was able to overcome, and she'll be telling us today about the behavior of highly pathogenic avian influenza virus in dairy cows. So over to you, Jan.
Yeah, so first I want to make sure like my slides read is good.
They look great, yes.
Okay, yeah.
All good.
Yeah, thank you. Thank you very much, Sam, for the introduction and the committee for invitation and this opportunity to present our research understanding the behavior of HPAI H5N1 virus in the dairy cows in this event. Okay, so like Tony gave a nice introduction on the HPAI H5N1 virus, and we know it is a subtype of influenza A virus, which has a genome composed of A-segmented RNA molecules. And influenza A virus is well known for its broad host range, infecting many species from the natural reservoir, which is wild aquatic birds, and virus can infect humans, pigs, chickens, other mammals. Interestingly, you may notice that cattle have been excluded. Historically, we know that cattle are known to susceptible to influenza D virus infection, which causes the mild respiratory symptoms, and it's usually self-limiting. However, in March 2024, there was a report that HPAI H5N1 in this clade 2.3.4.4b, and then genotype B3.13, this particular strain of virus infected dairy cow, particularly in the mammary gland. So early 2024, the farmers in the Texas area, they noticed that their cows experienced flu-like symptoms, including mild respiratory signs, lethargy, and decreased feed intake. They also observed a sudden drop in milk production, together with the changes in the milk consistency, as you see here, yellow in color and thick and curdled in texture. In some cases, the mammary gland involution is also observed. And laboratory tests confirm that all these symptoms is caused by high-path avian influenza virus, H5N1 infection. And up to May 14 this year, USDA has reported over 1,000 herds are infected and then spread in 17 states. So in response to this unprecedented spillover of HPAI in cattle, we quickly form a team. So we want to ask if experimentally we can reproduce the HPAI H5N1 genotype B3.13 infection in lactating cows. And we also want to determine to what extent the natural immunity confers the protection against disease and reinfection. So we brought in like 3 lactating cows, and we intramammary challenged into the hindquarters, in this case, into hindquarters, with 10 to the 4 TCID50 virus per quarter. The virus we're using is the Texas cow isolate. And 1 cow was euthanized on day 4, and the other 2 cows were re-challenged again through the same route, which is intramammary, and then with same dose of virus. At this time, we re-inoculate the virus in the front quarters, and they were observed for 10 days, and our whole study ends on day 14 post primary inoculation. So over this time, we collected milk twice daily, and then nasal swabs, urine, feces, and blood were collected on predetermined days. And what we observed that after the primary inoculation of the virus, cows all developed fever starting on day 2 and then resolving by day 5. They were all depressed and reduced feed intake starting on day 2 post-inoculation. And in terms of the hindquarters, the clinical mastitis was developed by day 3 post-infection. And the milk quality, if we see here, is the hindquarter milk is discolored and then is thicker and the consistency is changed. And in terms of milk production, we see a quick milk production drop starting on day 1 or post-infection and reached the lowest production on day 4, and then gradually rebounds. However, they never reach the original level, and so the milk production maintained at about 50% of the day 0 level. So after the re-inoculation on day 31, we didn't observe clinical signs, and there's no further milk drop. And consistent with this clinical observation, we could detect high quantity of viral RNA starting on day 2, and then peaked on starting on day 1 and then peaked on day 2 to day 4, and then slowly this viral RNA decreased until day 21. And then they maintain at this level with the CT value of about 30 or above. And the viral RNA was only detected in the hindquarters. And the 2 cows show pretty consistent RNA virus shedding pattern. And then we pick some days and to check whether what's the titer of the infectious virus. So we can see like on day 3 post-inoculation, the hindquarter milk has very high viral titer, reaching 10 to the 8 or above TCID50 per ml, and then decrease over time. By day 10 post-inoculation, we couldn't detect any infectious virus. And there's very low titer in the front quarter milk. Histopathology examination shows this on day 4 post-inoculation. The front quarter, the teat cistern and mammary alveola shows pretty normal. And then the rear quarters, which are inoculated by the virus, they're very severely affected by the acute necrotic mastitis with alveoli filled with the proteinaceous material mixed with the fat droplets. We often see alveolar epithelial cells slowed into the lumen. On day 41 post-inoculation, all quarters from 2 cows look pretty normal, And then we didn't detect any active cellular damage. The pathologists, when they examine the slides, they can tell there's a sign of healing. We also checked the viral tissue distribution. Here we appreciate that in the hindquarters, the high viral RNA level was detected. Medium level of viral RNA was detected in supramammary lymph nodes. And to the rest of the tissue we collected, and the body fluid that we collected at various time points, that include blood, nasal swab, urine, feces samples. The RNA level, their CT values are all in mid to high 30. That indicates there's little evidence of systemic spread of the infection. So we checked the systemic and local antibody responses. And here we see that in serum, both cows were seroconverted on day 7 post-inoculation. The viral neutralization antibody can be detected on day 7 post-inoculation, and then plateaued on day 14 post-inoculation. And the local milk antibody titers, we can detect both in the front quarter and the hind quarters. The total antibody can be detected on day 14 post-inoculation. In terms of the viral neutralization antibody and hemagglutinin inhibition antibody titers, they can be detected on day 14. However, you can see there's a greater titer in the hindquarters than in the front quarters. Seeing that the front quarter milk has lower antibody titer, which is only 1 to 10. And however, like when we inoculate, we re-inoculate the cow in the front quarter, they didn't develop any disease. So we're wondering the neutralizing capacity of the front quarter milk. So instead of running the conventional neutralizing antibody by incubating of 10 to the just 100 TCID50 of virus with the antibody, we increase the viral load. And then here we appreciate that the day 28 front quarter milk can neutralize up to 10 to the 5 to 10 to the 6 TCID50 of the virus. So that's showing this, though the front quarter milk had the lowest neutralizing antibody titers, they can completely protect from disease and reinfection. So we're also interested in identifying which activities have the potential to generate bioaerosols and transmission to agricultural workers. So here we strategically positioned 3 air samplers into the rooms. So we assigned 2 rooms to this study, and 1 room serves as the living quarters for the cows, and the other one is for the milking purpose. So what 1 air sampler is placed in this milking room at the level of the other, and the air sampler is turned on during the morning stripping milking and the mechanical milking, and then the second air sampler is set into this living quarter, which is 1.5 meter above the ground. And what happened is every evening, the cows are moved to the milking room. So this room is unoccupied. And then we power wash this room. So at this time, we turn on this air sampler to collect air for like 40 or 50 minutes. And then there's another air sampler is placed on the ground of this living quarter. That's after the evening milking finished, all cows are settled. So we turn on this air sampler for 3 hours and to collect air. And so here, and you can see that During the peak viral shedding in the milk time, that's on day 2 to day 4, we can detect some viral RNA, which are at about above like CT value above 30. So this indicates that the routine farm activities involving the H5N1 infected cows can generate the aerosol containing viral RNA. However, when we try to culture this, the eluent from the air samplers, we couldn't recover any live virus. So considering that our X3 animal room, the air exchange rate is 15 times per hour, this value probably be underestimated. So to summarize this part, what we observed is that primary intramammary inoculation with HPAI H5N1 genotype B3.13 led to disease that is similar to what was seen in the U.S. dairy herd. Thus, in terms of the clinical signs, viral shedding, mastitis in the infected quarters, and decrease in milk production. The re-inoculation of previously naive mammary glands does not result in the same milk drop-off. We didn't observe any disease. There's no viral shedding as initial inoculation. And antibody titers generated in milk is sufficient to neutralize the virus. We thought this is correlated to the protection. So all in all, we demonstrate that dairy cow developed the protective immunity against the reinfection with bovine H5N1 influenza virus. So now I show some recent data obtained in the lab. So we profiled the gene expression in the collected mammary tissue. And here I show that's the milk gene expression. This is a casein gene. This is a whey protein gene, and this gene encodes a lipid droplet membrane protein. So altogether, they make the milk. They contribute to the milk content. So on day 4 post-infection, the hindquarter milk genes are significantly downregulated compared to the front quarters, and they're pretty consistent across the across the genes. And then on day 41 post-inoculation, which is so the 10-day post the secondary inoculation, we see one cow, both hindquarters, and one of the hindquarter of one cow, their genes are further downregulated. And then interestingly, one hindquarter resumed to the normal level of function. So in terms of this quarters, we're not quite sure at this moment if the genes are permanently damaged or they need more time to recover. So we're also interested in see whether over this incubation time in the mammary tissue, whether this virus undergo further adaptation or there's any mutation comes up. So we send out we send our sample to Johannes and his group. So consistent with our results, they couldn't recover any live virus that's enough for RNA sequence. And so after 10 days. However, they could recover the virus from day 1 to day 7 and then got the sequence. So in the samples collected on day 1 and day 4, we didn't see any mutations in the viral genome. However, in day 7, there's a 30% frequency that the PB2 gene has 3 amino acid deletion, so as seen here. And then there's also low frequency that there's a point mutation on the HA gene. From another cow sample, PB2 has low frequency of generating stop mutation, leading to a truncated PB2 protein. And we know that PB2 is one of the RNA polymerase subunit. It is one of the subunit of RDRP. So we want to know whether this deletion has any impact on viral RNA polymerase activity. So in this mini replicon assay, we can appreciate that deletion of these three amino acids leads to the decreased polymerase activity. This is tested in mammary bovine mammary epithelial cells, as well as in human cells. So we think this deletion probably is detrimental or decreased the overall viral fitness. And then this probably contribute to the clearance of the virus and after day 7. We're also interested in seeing the mammalian adaptation that virus uses to replicate efficiently in bovine mammary gland. So we know that there's a single spillover of this virus from avian to cow. So originally, this is an avian virus. There's a well-known avian virus adaptation, avian virus that utilized to replicate efficiently in mammals. However, this bovine H5N1 virus, this PB2 gene, they still maintain the avianized amino acid on these two well-known positions, 627 and 701. So then we focus on the other two positions, amino acid at 631 and 649. So here, when we mutate the avian, here we thought, okay, this is a mammalian signatured amino acid, and we mutate that to avianized amino acid, and then we also make a double mutation here, and then in the viral polymerase assay, we can see that mutation of 631 from mammalianized amino acid to avianized amino acid, that's significant decrease the polymerase activity in bovine cells as well as in human cells. And this 649 amino acid mutation did not have much contribution. However, the double mutation confirms the key role of this 631 leucine that enables the avian virus to replicate efficiently in the mammalian species. So to summarize here briefly, milk genes expression in the hindquarters was downregulated on day 4 post-infection and did not recover on day 41. So gene expression in the front quarters are less affected. And we didn't observe any mammalian adaptation mutation in PB2 genes by day 7 post-infection. And PB2 leucine 631 plays a key role for this bovine H5N1 virus to replicate in bovine cells. So I will end here to acknowledge the incredible team behind this research. Tony brings in the expertise in bovine infectious disease and immunology. Bryce has a tremendous experience working in high containment labs. And we have incredible and talented research groups, technicians, students, postdocs, and even summer students, honor students. They work very hard and generate the data. Vito Veterinary Services did all the animal work with precision and care. Vito biosafety team ensures our procedures comply with regulatory regulations. Prairie Diagnosis Services, Yan Yun and his team did the pathology. Sam and Allison provide knowledge and air samplers, knowledge on air sampling, and then provide us the air samplers. Johannes and his team did the viral RNA sequencing. And then, of course, we acknowledge CIHR, NSERC, CFI Innovation Saskatchewan for financial support. All in all, we made this research possible. And thank you, everyone, for your interest.
And we certainly thank you, Dr. Zhao, for that amazing talk and for the really influential and pioneering global work that you and your team are executing at Vito with really unique models that are second to none. So really nice work, and thank you for that talk. I would invite all the panelists to turn on their cameras. We're going to go a little bit in question mode here, Q&A, to wrap up the session as we go. For Tony and Jan, it's going to be a little bit like a thesis style. Well, I'll be reading some questions from individuals. It'll take us back to our thesis defense, more or less days. Great memories, I'm sure. Just a general comment, a lot of queries on the board with respect to the 275 tyrosine mutation, Tony, and if other antivirals were tested, specifically peramivir or baloxavir in your assays, or if future queries on that will be tested.
So Balaxavir was not tested. It actually targets a different protein, so its efficacy shouldn't be altered by the particular substitution. I didn't mention, but they did actually test Paraminavir as well. It has some inhibitory effect. It's sort of intermediate between oseltamivir and zanimivir, which is exactly what's been published for other proteins with this particular substitution as well.
Fantastic. Thank you. Jan, can you comment perhaps if the cattle were fed poultry litter waste? I'm not sure if that was can you comment on that, Jan?
In our lab or in lab settings or in the field?
I guess both.
In lab setting, no.
In lab settings, definitely not. Okay.
No.
Tony, are you looking at reconstructing ancestral permissive substitutions outside of the NA and HA regions?
It's there. If there are mechanisms to do that in silico, I haven't been able to think of one yet. That's something we'll have to defer to the lab to do and do actual experimental work by knocking these viruses into cells and potentially into animals as well. But for the moment, it's tricky to identify with completely different proteins and completely different evolutionary histories. Trying to identify, trying to put together an in silico model of how these proteins interact is pretty challenging. At that point, it would be easier to do testing in vitro.
Jan, how significant do you think environmental contaminations are as a mean to spread the virus, be it birds, cattle, or other domestic animals? What's your expert opinion on that?
I think like in the case of this cattle infection, because there's tons of virus detected in milk, as we can see, like 10 to the 8, 10 to the 9 TCID per mil. So, and also when the cow are being milked, there's a splash, there's like in the field settings, there's splash, there's probably like aerosol or air droplet. So there's a lot of spreading of the virus, to the environment. However, in our settings, as I mentioned, like our lab is like airflow is very quick, air exchange 15 times. So we probably miss some opportunity to catch that aerosols.
Okay. Thank you, Jan. Tony, do you think it is feasible for epistatic interactions with M2 or other proteins? Any thoughts on that, Tony?
It is certainly feasible, especially given that HA and M2 they follow each other. So there is evidence that they do interact. So that is definitely feasible. Just we haven't got around to sorting out how and if we're going to test such a thing just yet.
Thank you. Jan, any plans to test the D1.1 virus in dairy cows?
Good question. So we've been thinking that, and now we're trying to develop a small animal model. I didn't mention this in my future direction. But right now we're testing. We're trying to do the like a goat lactating goat model. So we're testing D1.1 currently.
Okay.
Yes.
So there's a comment here about, oh, that moved very quickly, about GSK human H5 vaccine, Pandemrix, has been produced and distributed to the provinces. Perhaps Tony can field this one. Are there any triggers that are currently being considered to begin a human agricultural worker vaccination program? Is there anything you can say about that, Tony, perhaps, or an opinion?
I'm not sure on that. That's not something I'm directly involved with. Oh, I've just been told that no, not yet.
Okay, not yet is the answer. Thank you, Tony. In addition to your serological study, will the role of cell-mediated immunity be investigated also, specifically with respect to its role in protection to secondary challenges? So the question is about cell-mediated immunity for secondary challenges.
Is that for Jan?
Yes, for Jan, I
Oh,
apologize.
okay. Yeah.
Sorry, I missed. Sorry, I was reading like.
No
Sorry,
worries.
can you please?
So, if there's any ideas or thoughts about pursuing the role of cell-mediated immunity for secondary challenges, Jan?
Yes. So now we're analyzing the cell-mediated immune response. So we collected the leukocytes from the milk. We also have the PBMCs collected during this trial. So we're analyzing those and simulate with the H5N1 virus and see their cell-mediated CD8, CD4, CD8 response. So we're doing that direction, but we thought definitely like cell-mediated immune response will contribute to the protection.
Okay. Jan, do you think that inoculation of the virus via the mammary route caused systemic infection in the cow?
I don't think we didn't observe like virus spread to other organs. At least we didn't detect any infectious virus. We can detect some viral RNA in other organs or at very like CT values all above 30 or high 30. So I don't think there's a systemic spread of the virus. However, we do see some virus RNA in the supramammary gland lymph nodes. Yeah.
Tony, here's a question perhaps for you. Is there any historical evidence that the mammalian spread to the same extent would have happened in earlier flu pandemics, perhaps?
Yeah, I'm not aware of the actual proportions of mammalian infections in past pandemics like H5N2 back in 2014. Just off the top of my head, I don't think there were a whole lot of mammalian infections, and realistically, in the grand scheme of things, there aren't that many mammalian infections in this outside of the dairy cattle outbreak. It's a unique situation where it's farm to farm spread and just lack of containing the virus. It doesn't actually wildlife isn't really involved in the spread of that particular virus through dairy cow. There weren't any mammalian infections in the 2014 outbreak that I'm aware of, and mammalian infections are actually quite rare here in the grand scheme of things. They're mostly limited to mesocarnivores, things that would have been scavenging dead animals. It's maybe 5% of detections, and they typically don't spread forward because they aren't particularly fit in mammalian hosts at this point. And they tend to, at least in wildlife, they tend to die before they become fit.
Okay, you've done both exceptionally well, not only in your talks, but in your thesis defense portion. Just kidding. Great session. In the interest of time, we'll wrap up the questions here, and I will pass it to Dr. Mubareka.
Thank you very much, Dr. Dikiakos. And I have to say that is probably one of the most efficient Q&As I have ever seen. And I want to thank participants for being so engaged. Those are very thoughtful questions that brought out a lot of excellent answers. So thank you. Just before we close, I will make a shameless plug for the 11th International Symposium on Avian Influenza that will be hosted in Canada for the first time, specifically in St. John's, Newfoundland. So that's happening in June, if people are interested. And with that, I think we will wrap up. And I'm very grateful to Car, Dr. Dikiakos, Dr. Joe, Dr. Senor, and everybody who is involved, especially the participants. Thank you so much, everyone, and have a wonderful weekend. Bon weekend.